Okay, so it should now be on presentation mode. So I listened in a little bit on the talk before. Unfortunately, the Wi-Fi sort of destroyed my connection, and I really enjoyed that talk. So and I could see that there was some background to breast cancer. So I'm gonna skip a little bit of my introduction actually, because I think it's just a duplication. I thought it'd just be useful, just sort of put us on the map here. So we are in Bradford in North England, and we are very close to a national park, which you have got on the left-hand side there, Yorkshire National Park. And our institute is basically a purpose-built institute where we have everything from the design of new molecules to take new therapeutics up to a potentially phase 1 evaluation. So I was going to give you a little bit of introduction on the breast cancer, but I think you've heard a very nice background there from the previous speaker there. So I don't need to speak too much about it, but when we are talking about developing kinder treatments, smarter treatments, it's also about selecting the right population. So this is kind of a very simplistic view of the previous talk. But one that I normally give as a slide to people who are not so familiar with different subtypes. But of course, when we are developing smarter drugs, we need to try and target them to the different subpopulation of patients. So in this case here is breast cancer. And what we're particularly interested in our institute is looking at two, I would say broadly, type of therapies. One is looking at potentially proteins or enzymes that are highly expressed in the tumor microenvironment. And another one is looking at yet unproven therapies that are focused on basically providing a non-toxic therapy to stop the metastatic character of tumors at the time of surgery, where potentially cells can obviously survive and escape that. So if we're looking at, for example, systemic therapy, when in the case of breast cancer, you have got the clinical approved drugs, you need to then start looking at how you can target these more aggressive stages of breast cancer. And chemotherapy is still very much used in that space. I've put some examples of drugs there just below the red arrow on some of the drugs that are being used. We have also got targeted drug therapy. So these can include antibodies or antibody drug conjugates. And in principle, these are more selective for the tumor and therefore should also result in lesser toxicity. As with many drugs, even if they're designed for this purpose, there's still side effects associated with these. So we think that we can probably try and mask some of the drugs that we use and try and release these compounds selectively in the tumor microenvironment. And we work with clinicians to try and address this, of course. And I think sometimes it's a bit like a chess game in terms of what you do to the tumor. You know that if you give the patients, hormone-targeted drug like tamoxifen, what's going to happen next? If they relapse, then you have your next sort of generation of compounds that might be a little bit harsher, like the chemotherapeutic agents that will then destroy the dividing cells, etc. But can we do a little bit better? Can we help the clinician a little bit better by providing better therapies? And on the right-hand side there, you have the Trojan horse concept there, where you are basically transporting the soldiers into the heart of the battle. And in the context of cancer treatment, it's about, can we actually masking the cytotoxic therapeutics until they reach the tumor sites where they get activated. So again, the previous speaker spoke about the tumor microenvironment, and of course, it's extremely complex. I'll try and give you two very brief scenarios of how we could properly exploit the tumor microenvironment. We know that when we treat the local tumor, we will probably be able to reduce the tumor size. Can we completely eradicate the tumor? In some cases, yes. In most cases, patients might relapse. And then when the tumor repopulates and potentially metastasize, you have a much more aggressive tumor. So potentially smarter drugs could be included at the neoadjuvant stage or at the adjuvant stage to replace those drugs that are currently being used in the clinic. So we're particularly interested in some enzymes, and in normal cells, these enzymes are our friends. They do what we want them to do. But the enzyme expression in cancer microenvironment sometimes are hijacking these enzymes so they become our enemies, and that's not what we want to do. So we are interested in the enzyme expression in different cell types within the microenvironment. So MDR, multi-drug resistant cells, cancer stem cells or tumor initiating cells, hypoxic cells, and the proliferating cells are probably those that we can target already. So the first little snapshot is on treating hypoxic tumors. So we don't actually have any clinically used drugs to treat hypoxia. And there's been 8 prodrugs that have been progressed there, 1 from our institutes. And I'm going to show you just the concept of that. But the general problem, obviously, is when you've got hypoxic cells, these are furthest away from the blood capillaries. And let me just see, can I get a pointer option here? Okay, so you have got these cells here in blue. These are round about 100 micrometers or a bit further away, and these are already starting to have very low oxygen tension. They are very much like quiescence. They don't proliferate very well. They survive the treatment because we can't deliver enough concentration to these cells. Similarly, radiation therapy is not particularly effective in those cells because of the low oxygen concentration, and therefore generation of radicals that can then subsequently damage the DNA. Okay. So in our institute, we have been working on a prodrug that's gone into a phase 1, phase 2 clinical trial, but has not been progressed further because, unfortunately, because of intellectual property, patent has expired and is on a prodrug called AQ4N. You have it as a structure there on the top part of the slide here, and because of this negative charge here, it doesn't bind very well to DNA, but it's very similar to another drug called mitoxantrone, which is also used to some extent in aggressive stages of breast cancer in parts of a cocktail chemotherapy, if you like. So when you have this N oxide, it's negatively charged, and as it tries to bind into the DNA, it can't because it's repulsive to the DNA phosphate backbone. If you're looking at the same compounds, but without the N oxide, the charge at physiological pH is slightly positive. And when you're tracking these compounds in cell, you can see on the top image on the right-hand corner there, the fluorescence is out in the cytoplasm. The nucleus here is on not affected by the fluorescence, and the fluorescence is the prodrug. But when you then have the N or the oxygen on the tertiary amine there removed, the compound now binds very potently to DNA and inhibits topoisomerase II. And we know that under hypoxia, we can convert that N oxide specifically to the active metabolite, AQ4. So you have a selective mechanism of action there. And going back to the clinical trial, there's something that was done back in 2007, 2008. And the prodrug does exactly what you want it to do. So primarily, it hits the tumor, the hypoxic cells of the tumor, and can eradicate those cells and leaves normal tissues unharmed. Now, because this patent expired, we've been looking at some second-generation compounds, and we've developed them, and we're now progressing them, and they're doing also exactly what we want. So we have potentially metabolites like AQ4. These are covalently binding to DNA and damaging the DNA, so the cell cannot be, so the DNA can not be repaired, and the cell will die. We have equivalents that are prodrugs, and these are entering the hypoxic cores of spheroids. So on the left-hand side here, what you see in green cells are generally what chemotherapeutic agents are doing. They bind only in those outer layers of the spheroids and will kill those cells, but all the blue cells that tend to be hypoxic are left untreated. And our prodrugs penetrate straight into the hypoxic core, where they will be converted to a DNA metabolite that can then generate single-strand DNA breaks and double-strand breaks, as you can see in this COMET assay there. And then we've been working with Professor Stuart Martin at the University of Nottingham, and he's been looking at combining our prodrugs. So in the first instance, the prodrug we call KP167, and it does again exactly what we've designed it to do. And he's shown in combination with radiation therapy that actually this agent is an extremely powerful radiosensitizer. We published it recently in this paper below, and we are in the process of submitting our next paper. And there's something quite unique going on in that KP167 is a much better radiosensitizer than AQ4N, which was tested on patients. And we think that the underlying reason might have something to do with specific DNA damage response pathway defects that are present in some 2D and 3D breast cancer cell line models. Second part project is also using the enzymes, but no longer on hypoxic conditions. We are trying to utilize the same enzymes, but using a biooxidative mode of action. So we're using a sort of a compound or class of enzymes, a class of compounds called duocarmycins. And these you probably will be familiar with because they are very important class of agents that are now being attached to antibodies as a part of an antibody drug conjugate strategy. So we are doing both a prodrug strategy, and we're also doing an ADC strategy with these duocarmycins. So the duocarmycins, they bind in the minor groove of the DNA, where they covalently will bind to an adenine. This hydroxyl group there, I'm pointing out now, is extremely important for the activation of these compounds. If you remove this compound, it should not be able to generate the metabolite up here that can bind in the minor groove. So our hypothesis was, okay, if you remove that hydroxyl group in that pharmacophore, you should inactivate it. I'm not showing the data because we have published on some of this already, but by removing that hydroxyl group in this position there, it is no longer able to generate the metabolite that can damage the DNA. And there is 1,000-fold or more difference between this compound and the one on the left-hand side. So we're really deactivating, non-toxifying this type of agents. And then the question then becomes, can we use a P450 enzyme that naturally wants to hydroxylate a lipophilic compound or substrate and thereby activating it? And so, cutting a long story short here, just for the purpose of showing you the strategy, we have a lead compound over here. The hydroxyl group is missing here, and in the presence of tumors that are expressing CYP1A1, which is preferentially expressed in some tumor types, including breast cancer, we can hydroxylate it in the key position here, activate the compound, generate the DNA metabolite that will then create selective kill. What you're seeing here in the middle is basically an autoradiograph of a plasmid that we have radiolabeled to show that the compounds are active covalently binding sequences of adenine, as shown here. This is one sequence. So the prodrug itself, at up to 100 micromolar dose, which is extremely high for duocarmycin, there's no DNA damage. The activated compound, so a synthetic analog, is able to generate DNA damage down to about 100 picomolar. And if we take the prodrug and we incubate it with CYP1A1 and we extract the metabolite out, we get the exact same sequence binding scenario as with the activated compound. So that suggests that we are generating this, and we've proven this also using LCMS. So that what is really interesting about these compounds here is that they evade most known resistant mechanisms. So if you can deliver them selectively to the tumor tissue, they're really effective at destroying those cells. And for example, Here we've got a small panel of ovarian cancer cell lines, one that is resistant to doxorubicin, one that is resistant to cisplatin, and they remain extremely potent. So these are picomolar IC50 values in cells. So they are, even though slightly affected by maybe some potential resistant mechanism in these cells, they remain picomolar active. But the in vivo study I just show you now is using CHO wild type and one that is transfected with the enzyme that can activate it. And I'm showing you the data set here. So we know we can get the activation by 1A1, and now I'm showing you in vivo coming here. This is a single dose just as a proof of concept. So the top graph there is untreated tumors. And if you treat it with a single dose of either the prodrug or the activated metabolite, we get actually very similar tumor growth delays. Where the big difference is when we're looking at the body weight, so as a form of looking at toxicity, in red here, you've got the untreated tumors. In orange up here, you have those that are treated with our compound, our prodrug, and those that are treated with the cytotoxic analog. And the one that is treated with the cytotoxic analog are clearly toxic to the mice. And 4 of these have been tested on patients as cytotoxic agents themselves, and they failed in clinical trial because they were too toxic. So the therapeutic index was too narrow. So we are showing that actually we can deliver this selectively to the tumors using our strategy here. And the same tumors, so the CHO cell line growing up as a tumor here, not expressing 1A1, when we treat it with our prodrug, we have no tumor growth delay. So that shows you the importance of having the expressing enzyme in that tumor. Okay, so we can safely dose with our prodrug, and we are now progressing this in these 4 cancer types here. So in addition to breast cancer, we are also shown in vivo we can treat tumors that are expressing the enzyme in bladder cancer, colon cancer, and head and neck cancer. Okay, so this was one example. We're also looking at another enzyme called CYP2W1, which is highly expressed in metastatic colorectal cancer cells. And we have shown this in vitro and in vivo, but I'm not showing you the data here. There's some more publications coming on that. Just remains me to say thank you to all my the people in my team, national and international collaborators, and to the funding bodies that have supported this work. And thanks to all you listening here, and I'm very happy to take some questions.